So we are looking at how do we, from with our expertise, get a particular author in there. We will ask the president to give a talk. What are the mandates of this institute? What does he have in dream as a dream for collaborating with community? After that, we will deliberate how we take it forward. They will become Das is in a FRC meeting. And once he finishes,
Governor Maniston, she's coming with the
Dr. Srikanth is coming. The Fulbright scholar.
Okay.
Big Srikanth.
So all of them, they are
full big connection of CC. Yes, sir. Srikanth.
all of them, they are now from different laboratories.
You know that? Pooja? Why don't you introduce biotechnology people?
Yeah, sure, absolutely.
Pooja is now.
I know Pooja.
Yes.
Introduce your colleagues, new
Right.
colleagues who have joined.
So from biotechnology, Dr. Seema is.
I don't know her.
Yes, and then we have Dr. Rashri.
Yeah.
Dr. Rashri Das has also been with
She
us.
works on
for many-
HPILO, right?
Yeah.
Yes, sir.
And she.
I know her from her son, who's a good football player. Yeah. He's in 12th now, or which class? Ninth class. Yeah.
And then Dr. Shruti Khanna, she's just joined AIB, I think 6 months back.
Yes, ma'am.
And Dr.
You are teaching or you are in research?
I'm to teaching and research both.
Yes, everybody's on teaching or research. And Dr. Balashri.
Okay.
So Dr. Balashri has also joined, I think, a year back,
No,
around 8, 9 months.
You have some
8, 9
international
months.
experience? You have some international experience? Have you been working
Yeah,
on
they both have postdoctoral.
Let us speak.
Can you please introduce yourself?
yes, so myself, Dr. Banashri. I joined Amity on in April 2023, and I recently got one project also of ICMR DHR, and that is with natural hybrid products. The role in breast cancer with few genes that I am not disclosing right now because I have selected those genes on the basis of whole exome sequencing and NGS in our Indian population. And other than that, I'm also interested upon some research work on oral contraceptives also because oral contraceptives
additional
have
chairs
great
carry,
impact, both health, mental health wise and physical health wise in women, and it has environmental impact also because if you're taking it randomly, it is releasing in our environmental waste also, and that is causing havoc in our aquatic natural system and soil natural system. I'm planning to do some detection kit that is in my near future I'm planning for, and I have submitted few projects regarding that, like in SubSure and in CSR Aspire also.
But have you been working with some international universities or collaborators also?
Right now, I'm looking for such kind of collaborations with whom I can done,
Okay.
but I have a few collaborative work with the ICMR field.
If you can, I don't know whether you know me. I'm Gurinder.
I know you.
Yeah, and I am a group vice chancellor also. So maybe if you can send me an email, what are your research areas? And then Dr. Pooja, if we can get to know that which are the major strengths. So for example, as a biotechnology, which are the major areas? Well, people keep on coming to us, then we can relate you.
Yes.
Also, do you know about the study abroad program which we run?
Yes. A few of my BSc students has gone for a SAP program, and they came back from there. 2 of them just came back from there. So I know a bit of these things which in Amity is going on. And I just know you from the background of yours, as you have done your MBA from IIFT.
Yes, yeah.
And
That is from IFT?
no, I'm not from IIFT.
Yeah.
My husband got opportunity in IIFT and FMS. So he did his MBA from FMS. So in that
Okay,
context,
so maybe that he knows me.
IIFT.
Right. Great. What is your husband doing now?
He is in government consultancy, policy making and all. So he is associated with a consultancy firm. Earlier he was in KPMG. So he knew Amity because lots of students from Amity used to go to
No, KPMG are partners. I do a lot of projects with KPMG. So maybe that we can then be associated on LinkedIn. All of you, I would request that if you are, I'm quite active on LinkedIn. So if we can get to know each other through LinkedIn, because I think there is not a day when we are not coming out with one project or the other. But very warm welcome to both of
Thank
you.
you so much.
Yeah. Dr. Pooja, I'll tell you that one of your students, his father is a CEO of a pharma company
Okay.
at Ahmedabad. So one day on Sunday, I got a call. So this was a Sifarish call from someone. So my yeah, my wife happens to know this lady, and she says that and then she says that I have a little recommendation to be made. Tell me, and then she
Kirana
says
loud
that,
here लिखे.
you have a study abroad program, and from biotechnology. This is a very bright student, by the way. And I want that
Already started.
Engineering.
he is studying with you, but he has no clue about what study abroad program is. And so she was saying कि मेरे बच्चे को भी लंदन भेज दो. So मैंने कहा बिल्कुल भेज देंगे तो anyone can go, there is no problem at all. But I think that maybe if all of you, whenever you get some time, if you can speak to them, that would be great. But very warm welcome to both of you. I think that our faculty is doing a great job whenever they are abroad, but we will introduce you with the help. Who else is there?
So,
Shruti.
Dr. Shruti is there.
Shruti, I just dropped. Yeah. Tell
I
me.
did my, I'm basically a chemist and I did my graduation and post-graduation from Hansraj College, Delhi University. Followed by that, I did my PhD from IIT Kanpur.
Which year you were in Hansraj?
I was, that will reveal my age. It's long back.
I'm actually from DU. That's it. Yeah.
I'm also alumnus of.
I was at D school. So I was at D school, big
Okay,
school, at D school. So we have got very close association with Stephens Hindu because they were our neighbors, and then Karodimal, and then they took the Hansraj. But you have a, but you used to have a festival called Renaissance, I think.
It was confluence.
Confluence.
Hansraj.
Yeah, I used to be a debater at my time. So there was a Mukherjee Memorial debate where Hansraj debaters
तो
were
है
very
ना
active.
यहाँ रखते हैं ना.
So that's why I'm asking that which year you were. But don't tell me.
The science part was very,
But we can.
laid back and it was never highlighted. Of course, academically
Let
it was
those share,
highlighted,
yeah,
but
yeah.
otherwise in all the other activities it's not little.
You can make it comprehensive.
So yeah, followed by that I did my PhD from IIT Kanpur in the field of bioorganic chemistry with Professor Sandeep Verma, and then I moved to IIT Delhi as an NPDA fellowship by Sir DSP, and I worked there in the field of biomaterials, and I continued working there as an institute postdoc also. So I had a total tenure of 5 years there, and my research area focuses on biomaterials specifically, and I'm also targeting AMR, antimicrobial resistance, where I have worked in the field of theranostics as an antimicrobial as well as diagnostic tools. So we have kind of made small rapid bedside assays which can be utilized in hospitals itself for the immunocompromised patients and the hospital-acquired septic infections, right? Most of the people we know, we come across that they have gone to the hospital for some disease, but they end up saying that it's a multi-organ failure. So it's basically a bacterial disaster that is happening inside the body, and because of which, so there's a rapid urge to diagnose such infections, and we're working on that.
Great. Thank you. Is there anyone who is working on climate change?
Kamal, acho.
Climate change. You are working on climate change? But are we running some programs on climate change now?
No, currently there is no program. We did short-term FDP program on that.
And are we doing something on sustainability?
नहीं.
Sustainability, we prepared the
No,
program.
but we prepared, but have we launched finally? We have not done it.
We should launch as soon as possible. I shared the whole syllabus in
Yeah, but clean air, change, climate change, we have actually not done much work.
We have our own air pollution monitoring station. So we have PhD students working in this area.
Okay. Climate change पे करते हैं?
Air pollution, climate change,
Okay.
and impact on cognitive behavior.
Okay, so they are working on you or?
100%.
And you, who's heading environmental sciences?
Environmental science joint coordinators were there, Nandilava and Renu Dupar. And now Renu Dupar is looking after it.
But who's the domain head? Who's the domain? You are.
And research I am heading and academic doctorate.
Dr.
That SP thing is looking up.
थोड़ी सी email कर देना.
Because for SAP, he can help
No,
you.
no, not that. I'm talking something else. Well, we want to understand who's doing climate change, who's doing environmental science, and who's doing sustainability and green energy management. So there are few projects with whom I think that we need to be associated few people.
For green energy, they are working, they are using algae for biodiesel production
Okay,
with nanocatalysis.
right.
So that we are working.
Okay.
So some projects, some people are there interested.
Yeah, actually, there was someone from abroad. They visited us yesterday. So they were talking about these 5, 6 areas. And they said that how good MET is. So we, as usual, we said that we are very good. Our people are doing. We took your name also. But then we wanted to understand that what all is actually on ground. So I think that clean uh, air and climate change. These are the areas where the government will be giving huge funding. So we need to make these departments strong. But you are the only one who works in this area.
Me and my whole group and some PhD scholars are there, but we would be needing some more faculty who have expertise.
But presently, only 3 people are working. Reliance is coming up with huge funding. Adani is already into it.
Okay.
Yeah, so I think that we need to understand that where the
In fact,
government is
engineering
focusing.
people also can contribute because they are looking for engineering solutions for clean air, technical solutions.
Okay.
For example, rising towers.
I checked up from engineering department. I think there's no one who's working on the clean air management. From at least from MIT, there's no one. So these are the areas, right? You can look at.
We've been working on ozone.
Public health people also are there. Who are
Okay, who are the public health people?
Regina and Shanawaz.
Yes,
Can you just introduce yourself to Group Vice Chancellor?
sure, sir. So good evening, sir. I'm Dr. Shanawaz, Mohammed Shanawaz. So basically my after that I did my MPhil and PhD in population studies from International Institute of Population Sciences in Britain. So my research is basically interdisciplinary on the crossroad of public health, statistics, and development. I also, as a in the part in the collaboration of GBT, Global Burden Project by the University of Chicago.
Wow.
Here I am working as a collaborator since.
Visitors are in.
So with that collaboration and also with the other project, there are many publications.
Sorry, what you were doing in University of Chicago?
So there's a huge project, GBT project, Global Burden of Disease project. Global Burden of Disease. So what they do, basically they collect the data from various countries, almost every country in the world. And although in spite of being deficiency in the data, like many times, many diseases, they don't have the small scale, small area estimation. Also, the many diseases are not available at all from some in means as a records are there or something. So, but with the help of various techniques and also with the help of their very robust and very complex mechanism, they try to give estimates on the world level of the various diseases, also the changing pattern, also the various factors. So I am engaged in that collaboration. So in that collaboration, I have many publications in good journals like Lancet, Nature, and all that. So with that, I'm working, and also
And these are when you are contributing, you're writing alone or you're writing with some.
So in the group, so in the groups, there are many collaborators from worldwide.
And you write collectively.
Collectively. So after the collection,
Meet me sometime. I'm quite actively writing, but I give economics or management angle to the issues.
Sure.
Right. What is your H index?
My index is presently 37.
37. Very good. And how many citations you have?
More than 21,000.
Wow. He's a good person.
Mm.
Right. Please meet
Yeah,
me.
you are
Meet me
here.
every week.
37 is fantastic.
Sure, sir.
Right. Great. Yeah.
Shazina, you know.
Good to see you. Truly good to see you.
Thank you.
Shazina, you know him.
Right.
Good evening, sir.
She also has nature genetics one paper.
Right.
My name is Dr. Shazina Saeed. I am a clinician by training, and then I did my specializations in population genetics and clinical genetics from University of Pittsburgh. And then I joined Amity, and I'm a public health person trying to look at the genetic landscapes. So that is what I will probably talk about today. And
Very nice to meet you. My pleasure. Thank you for taking time.
It's our honor and privilege to host you here and then have you.
Well, I look forward to discussions with you.
Hello. Hello. The long travel from.
I know.
Reporter from US.
Hello.
जी बिल्कुल
Dr. Richard, what a pleasure to welcome you. I met you in Colombia.
Yeah.
Long time back when you made a presentation.
Sure.
Yeah. It's such a pleasure to meet you.
It's great to see you here.
Yeah, absolutely.
About him I was talking to you.
Yeah. Great. Please.
Okay, good. I think. Put my bag up here and take my coat off. Okay.
ये तो आपकी जगह है मैं यहाँ आ जाऊँ बीच
Wet my whistle here.
So was it a long travel from Gurgaon to this place? Was it?
It's a little bit of traffic. I suspect that you're probably used to this. And I've lived in many cities in the United States. Traffic in San Francisco is probably about as bad as you have here.
North Carolina too several times.
North Carolina
drove
traffic can get bad too. So. Oh, thank you very much. I'm actually out of cards. So. Oh, actually, wait a minute. I do have, wait, hold on. I think I have two more. Yes. Two more cards.
One was kind, she called us.
Thank you very much.
I should be part of the meeting.
Thank you very much.
So shall we start the meeting? If you okay with it?
Absolutely. Let's start.
This room or washroom.
I think I'm fine. I think I'm fine.
So first of all, let me thank Dr. Kamal Rahman to bring such a wonderful guest who is a great accomplisher and who has contributed immensely in the field of environmental health. Dr. Rick Woychik. And I had the opportunity of visiting his institute, NIEHS, 3 months ago. And when Dr. Linda was just handing over the charges 4 months ago, I visited. I was able to see around all the wonderful work that's happening in the institute and also the plans which you have for the future. So there are many commonalities between what Amity is doing. There are many areas in which there are convergence of our interests, competence, and also our future goals. Converge exactly in your area, in the National Institute of Health and also National Institute of Environmental Health Sciences. When I looked at your profile, which I was just talking to my colleagues before you arrived, here is the person who has got industry experience, who has got laboratory experience, who has got the leadership experience in different domains. So I was fascinated right from your early education, which you had, and postdoc. All this shows very high competence and significant impact which you have made in the field of environmental health. So it's a great privilege to work with here. And he is now the director of this great institute. And prior to that, he has also been deputy director over here. And so his main area is gene cloning. One area where I was fascinated since I have worked in that area is on hypothalamus, where the feeding center and satiety center, which is how does it get affected in high altitude? When the soldiers go to high altitude, they don't feel the good appetite. So their appetite goes down and they lose body weight, 6 to 8 kg when they stay for 3 to 4 months. So we were looking at the neurogenic centers which regulate the feeding and satiety. And we were looking at leptin, neuropeptide Y, all these hormones. So when I looked at your work,
Yeah.
which you have cloned a gene which is actually looking at satiety and correlating that with obesity. See, in obesity, people keep on eating without any control. So there is this satiety center and the protein which is regulating the food intake and giving the signal of satiety is abnormal in this case. So he has cloned that gene. So that was some fascination I had when I looked at that. Then the other area, we have a very strong environmental group here. We have Amity Institute of Environmental Sciences, Amity Institute of Environmental Toxicity and Management. Then we also have Amity Institute of Molecular Medicine and Stem Cell Research, Amity Institute of Biotechnology. We have a strong bioinformatics group, and we have a strong cancer group because you have worked on cancer as well.
That's right, that's right.
And we have a
Director of the
very
Cancer
strong,
Center.
you are director of that cancer center. And we have all cancers, right, from breast, cervix, head and neck cancer, oral cancer, and also liver cancer, pancreatic cancer, in all these areas, we are also working. So we have that group, Amity Institute of Molecular Medicine and Stem Cell Research. We have Amity Institute of Public Health, which is very important when you are looking at the environmental health, the hygiene, and also climate change, how will it affect. So we have a very strong Amity Institute of Public Health. And we also have a strong AI group. We have Artificial Intelligence Center of Excellence we have established here with a high computational capability of 10 petaflop currently, and it is going to be augmented to 60 petaflop. So we can look at predictions, projections, how these, whether it is environmental health, how will it change in our psychosocial environment? What is the pollution level which we have? What are the biomarkers which we can use? What are the epigenetic factors which we can use? So all these areas we have tremendous strength. So it is Dr. Kamar Rahman, we discussed with her that we should now, this opportunity when you are here, we will look at how, what is the specific area of your interest where there is convergence between IT and your institute. So I welcome you and we have founder Dr. Ashok Chauhan, who has created this whole Amity University with 12 universities in India, 18 campuses abroad, 28 schools. We have 200,000 students, 10,000 faculties, 5,000 PhD scholars, and we are focusing on research and innovation. And we have filed more than 2,080 patents, have published 35,000 research papers, and our H index today is 138 university. Our ranking at the national level is 22. So this institute is the university in the last about 10-15 years. They have done a remarkable job in the field of environmental health, which you will hear from all my distinguished faculties here. And Dr. Ashok Chauhan has sent his greetings, best wishes, the founder president. And I was also told that you had a wonderful lecture there at Amity University, Haryana. So now, with all that you traveled all the way to reach here and then share your thoughts with us, how do we build partnership with you? So I welcome you. Just to give a little brief about his profile. See, he is the director of the National Institute of Environmental Health Sciences, which is a part of the NIH. And he has done his PhD in a very important area and MS and PhD from Case Western University, Case Western Reserve University in 1984. And his BS and MS is from the University of Wisconsin-Madison. And he did a postdoc from Harvard Medical School. And so he has got a lot of industry experience from Jackson Laboratories. He has been the CEO of that. And he has also been with NCI Designated Center, Cancer Center. He has been the director of that. So he has a very, very strong, and he is now the director of a national toxicology program. So such a great distinguished person is here with us. So let's all give a very big applause to him. And we have very senior officials like Group Vice Chancellor Professor Gurinder Singh, and who looks after all the campuses and also internationalization as a focus. So may I request you to say.
Thank you, Dr. Silamurthy. What a pleasure to welcome you, Dr. Richard. A week back, Dr. Kamar Rahman, who is a very accomplished scientist. She called her and she said that she is going to bring a very important person to MIT, and we had a long chat. And I think that everyone here is looking forward to understand from you that which are the areas we can work together. MIT is a dream child of our founder. But what is great in this university is that the research is in our DNA. And we want to, yeah, and that's
You
what.
said all the right things.
Yeah.
Research and DNA. You got my attention.
Yeah, absolutely. And in this room, there are researchers who have got not only very high research index, but they keep on working on very exciting things. When I was sitting, I was just talking to four of the newly joined faculty members. Brilliant. I think that the way they are working. We are also looking for creating something in climate change. And on ecology, I think these are the areas where, when I was looking at where your specialization is, India is typically a country where environmental sciences, especially climate change and green technology management, are the areas where the government is focusing on. MIT is trying to understand that how we can support government in these areas. But we look forward to understand from you that how we can work together. Thank you so very much. Once again, very warm welcome.
I would request Kumar Rahman to say a few words and then we will listen.
Yes. Thank you very much, Doctor. Thank you very much, Dr. Sigamurthy. And I'm very happy that today Dr. Rick is here. I'm associated with NIHS from 1979. We had organized an international conference. In that conference, 27 countries participated. It was on environmental and occupational lung diseases. Then second conference we organized in Lucknow again, and it was impact of environment on women's health. Yes, you was there. And that time Linda was there. And then we signed a MOU between Amity University and NIHS. Now we have to renew with you. And I'm sure that your interest is the impact of environment and impact of climate change on human health. So we will organize another meeting and we will have a very nice collaboration together because it's a very important subject and especially for vector-borne diseases which we are facing in India. So I will not talk much. I welcome you.
With this, may I now request Dr. Richard, what are the dreams you have? What are the areas you feel that you are looking for a collaboration, partnership in India and with MIT? So over to you.
Sounds good. Well, I'm thoroughly, well, first of all, let me thank you for this invitation to be here. And actually, I specifically want to thank Dr. Raman for the invitation and the encouragement to come and visit. So thank you very much. I'm just blown away by learning about all of the different things that you're doing. So, as I mentioned this morning in my presentation, innovation is in my DNA. And innovation and collaboration, and a lot of innovation comes out of collaboration. So I really look to build collaborative opportunities where you bring something to the table that's different than what I can do, and I bring something to the table that's different than what you can do. And if we're both interested in, say, climate change and health, we can then figure out, well, how can we be working together? And when we develop these productive collaborative relationships, we can take science places where it wouldn't have gone if you're doing it by yourself or I'm doing it by myself. So I think that's the critical thing we want to look for is, how can we be collaborating? And not just collaborating by talking about collaborating, but really coming up with some fundamental things that we can be doing together. So why don't I talk a little bit about some of the areas of scientific focus. So right now, we're engaged in a 5-year strategic planning effort. So we're looking out over the course of the next 5 years. And in fact, I have a set of slides, which I'd be happy to share with you. At, well, I don't have them readily available with me. So let me just kind of speak at a high level, but I'd be happy to provide you with a slide deck afterward. So there are 6 different areas of primary scientific focus that are emerging as part of our strategic plan. mean, the first area of scientific focus is really to begin to embrace what we call the exposome. So many of you remember the Genome Project, right? So the Genome Project was all about, this bold vision to look forward, that if we truly want to understand human health, we have to get beyond studying one gene at a time. And I totally understand. As my graduate work, I actually cloned the growth hormone gene. And so we thought, well, you clone the growth hormone gene, and you can extrapolate out and know everything you need to know about human biology. Well, you can't. So the Genome Project was about going beyond one gene at a time. And that's been enormously successful. So it required that we developed new technologies. Who would have thought back when we first launched the Genome Project in the 1980s that you'd be able to sequence an entire human genome? Now you'd have RNA-seq, and you'd have some of the powerful tools that we have available. Well, that all arose because of this powerful vision, and there were technological developments, and now we're at a point where we can actually deliver on that original vision. Well, we want to do the same thing with environmental health sciences. So if we truly want to understand the impact of the environment, we can't study one exposure at a time. But the exposome is a research framework where we can begin to look at the totality of environmental exposures. So it's not just about PM 2.5. I mean, ozone plays a very important part of air pollution. And we have pesticides. We have multiple different kinds of pesticides. Actually, in my own laboratory, we study actually the effect of rotenone, which is, tickling complex 1 in the mitochondria. But it's not just that one pesticide. So we have to study all of the physical chemical elements of the environment. And it's going, just like the Genome Project, it's going to involve developing new technological and innovative new ways to study the environment. And some of this may be developing strategies to look, say, at the epigenome. So and we actually know in my own lab that it's not just the mutations in the DNA, but epigenetic changes in the genome can be influencing how your genes are working. So there may be epigenetic marks that are reflections of complex and interacting environmental factors. And we need to sort those out. So that's all part of the Exposome project. But I also want to point out that I think we all understand that it's not just about the physical chemical exposures, but sleep, nutrition, psychosocial stress. Those are environmental features that we have to integrate into the way that we study the environment. So are there, for example, epigenetic features of psychosocial stress? What about sleep? Are there epigenetic features of sleep? And so the Exposome project is really about integrating all of these different features together and coming up with a new operational definition of how do we do an exposomics experiment. Actually, Dr. Collins, many of you probably know Dr. Francis Collins, who is the former director of the NIH. I talked to him a lot. He said, Rick, he said, look, I'm a geneticist. How do I do an exposomics experiment? I get it. The environment is really important. So how do I do that? Well, that's our responsibility. We have to figure that out. It's just like back in the 1980s. How do you do a genomics experiment? We didn't know how to do that. We had some of the early phases of the technologies. Does anyone remember the cDNA clones on microscope slides? The very simple microarrays. It was the first phase of doing genomics, but we're in a much better place now with RNA-seq and some of the other remarkable capabilities that we have to the experiments. We need to do the same thing with environmental health. And that's really embracing the exposome. So we have just launched a center, and we will be announcing the awardee. The grants came in. We're doing the peer review. So we will award one center, and that one center's responsibility will be to work with the global community to develop the operational definition of how do you do an exposome experiment. So stay tuned on that. And you can be helping us with this. If there are ideas that you have about exposomics, we'd love to be able to hear about this. Now, another area of great interest is climate change and health. So I think you all realize that President Biden is someone who doesn't ignore climate change. He actually embraces the fact that we need to do something about climate change. So as soon after he was inaugurated, he issued Executive Order 14008, and that was specifically to address the effect of climate change at home and abroad. And he pointed to the NIH, and he pointed to my institute specifically and said, Rick, we need a strategy to study the health effects of climate change. And there are many direct effects. It's remarkable how little we know about the biological effects of extreme heat on human physiology, and the biological effects of extreme heat on pregnant women, on children. So there are so many things that we don't know. Better understanding the biological effects of smoke and cardiovascular disease. So there are so many things we need to study. But there are also indirect effects on health and climate change. So if you have a hurricane that comes through and it wipes out an entire medical capability, so there are going to be patients who are looking to their dialysis centers. The dialysis centers are not going to be there anymore. So there are the indirect effects of climate change. So we have put together a strategy to address the health effects of climate change, and the URL is nih.gov/climate and health. That's one word, climate and health. So if you're interested in looking at the details of this, take a look. So Congress gave my institute, we're about $1 billion a year. So we're not the biggest, we're not the smallest, but hey, I'll take $1 billion every September 1st. It's in my back pocket. And the key thing is to figure out how do we spend that money in a way that we can get maximum benefit for the dollars that we have. So we had $40 million last year. We'll have this continuing on. And we're trying to figure out, can we use that money? 40 million is not a lot of money, so we're using it in a way to catalyze cross institutional collaborations. So I've actually reached out to several other institute and center directors across the NIH to develop a collaborative relationship. So Gary Gibbons, who's the director of NHLBI, Heart, Lung, and Blood. Diana Bianchi, who's the director of Child Health and Human Development. Josh Gordon, who's the director of the National Institute of Mental Health. So we're all working collaboratively together now, using that 40 million to catalyze collaborative relationships. And then, each of our institutes can toss in some additional money to help us fuel the necessary research in the area of climate change and health. So another area that we're very interested in is when we do toxicology testing, it's kind of getting beyond just injecting different amounts into outbred rats and counting lumps and bumps and deaths over the course of time. We really want to start focusing on mechanistic toxicology. So if a given chemical is, say, a class of perfluorinated alkyl substances, if it's causing biological effects, how is it causing biological effects? What are the molecular mechanisms? We could use animal models to study this. But if you begin to understand the molecular mechanisms of why animals are getting sick, you can take that knowledge of the molecular mechanisms and translate it to humans. That's what we want to be able to do. So another area that we're interested in is environmental justice. It's really beginning to study the impacts on communities that are most impacted by environmental exposures. These are the communities that are downwind of lead smelters. These are communities that are located next to toxic waste dumps or Superfund sites. So we're actually reaching out and listening to what their concerns are. And then doing the type of science and developing the type of data that we can take to the EPA and to other places so that we can fix their neighborhoods. And I'm getting on in front of these groups and I'm listening to what they say, and they're actually pretty angry. Okay. We've done a lot of research in many cases. We know that there are cancer clusters and that they're being exposed to toxic chemicals. They want their neighborhoods cleaned up. So we want to help them develop the data, which we can take that to different regulatory agencies and help them clean up their environments. So why don't I stop at this point? That'll give you a little flavor. I mean, the other areas would be data management. So if you're going to do exposomics and you're going to do, actually the other thing I should talk about is precision environmental health, which is really precision medicine with an environmental element to it. So taking a look at someone's genome sequence and then adding environmental exposures so that we can do gene by environment experiments on a genome-wide scale. That's another area of focus. So then managing the data associated with all of this is another area of focus. So 6 primary areas of focus. I'll share my slides with you. You can take a look at it. And in a couple of months, we'll be publishing our strategic plan. So that you're going to read all about the 6 things that I just talked about. So I don't know, does that give you a little sense of what we're doing?
Absolutely.
And I suspect that just listening very briefly to your focus, that there are many different, there will be many opportunities to collaborate very effectively.
Thank you.
Questions?
Thank you, Dr. Woychik.
Yeah, so my lab works on multiple projects. One of the area which is very close to exposome is in 2016, we published work human obesity network map where we screen millions of articles and we picked up each and every molecule protein which also had a body which you have mentioned. And we made a system network. And that was one of its kind. It was published in PLOS One, and that is being used by researchers. Now we want to take it further, integrate the environmental effect, how sleep, how eating, and how food and other activities is doing. We want to take it to the clinical side. Being a bioinformatician and an AI expert, I'll be happy to contribute in this exposome. The second project which I want to discuss.
Can I just make a comment before you go on?
Sure.
So having someone working with us that has those capabilities of bringing AI and ML. So we have data sets now. And we have data sets, gene expression data sets. We have whole genome sequence data sets. And once we operationalize the exposome, we will have then exposomics data sets. But the key to understanding the impact of the environment on human biology is integrating across all of those data sets. I mean, right now, what we do is we take one data set and we correlate it with human biology, genome sequence. We take another data set, transcriptome analysis, we correlate it with genome biology. We need the capabilities then to integrate across all of these different data sets. That's where you can help.
Yeah.
I'm not a bioinformatician, yes, but we need bioinformaticians.
Okay.
Who can look at the multidisciplinary, multifactorial.
Correct.
So one solution which can give you right away is a multi-layered integration of these, coming from genomics, proteomics and all, and they are all interacting with each other. So that can give you a right kind of answers, what genes, what pathways, what networks are affected, how molecular mechanism is affecting, and how the sleep and other networks are correlating and interacting with each other.
In fact, just along these lines, you can pay attention to the fact that we are currently engaged in a collaboration with NHGRI, the National Human Genome Research Institute, on developing innovative strategies, AI and ML strategies for integrating across multiple different omics data sets. So pay attention, that's coming down the pike.
I'll be absolutely interested.
You're probably aware.
He's working with Carnegie Mellon.
Yeah, I'm also working with Baylor College of Medicine. So that is the second project which we are accelerating the vaccine development process. Normally a vaccine take 10 to 15 more than a couple of $1 billion to get into the market. So what we have built AI-based platform which cuts down the cost of vaccine development, and it also speeds up by at least 30 to 40%. Again, we have integrated the network medicine, network biology, and artificial intelligence systems, and that has been, our predictions are being checked by Chagas disease use by the Baylor team. That is going to enter into phase 1. And our team in Baylor got one vaccine in India for COVID-19, and they sold 100 million doses in that. That is open vaccine for all. Like, it's a patent-free vaccine where we don't charge anything, and that's a free thing which we have brought to benefit the masses and poor.
So here's a challenge for you.
Yeah.
So right now, the way we do vaccine development is that we essentially do it on a cross-section of the population. Some people may be more immunosuppressed than other individuals, but we develop the vaccine as effectiveness across this highly diverse population. So using the concept of precision environmental health, precision medicine, I mean, a challenge for you in vaccine development is, can we now start looking at individuals as part of your clinical trials? Because the dose of the vaccines for different individuals to be equally effective may be different. So just something to keep in mind. And actually, this goes back to some of my, as you indicated, that I spent time in different sectors. So back in my pharmaceutical days, there was a lot of interest in, this precision medicine. Trying to identify the individuals within populations of, say, type 2 diabetics that would uniquely respond to a given therapeutic. And then other individuals that have the same phenotype, type 2 diabetes, but are developing the disease for different molecular reasons. They may be then responding with a different therapy. But anyway, getting back to vaccine development, we know, and I know that those of you who study the environment know that there are things, perfluorinated alkyl substances. I mean, one of the principal things they do is they suppress your immune response. So, so do we need to then, if we knew that ahead of time, that there were individuals that may be genetically predisposed and who have also been exposed to PFAS, then the dose of the vaccine may be different. And, the beauty of the mRNA vaccines is that you can now adjust that pretty quickly. So just something to keep in mind as you continue to
Just to
advance.
supplement, we have established a very strong center of excellence in AI with the 60 petaflop computational capability. The first phase 10 petaflop is functional now. We are upgrading to 60 petaflop, and we have established a data science center, bioinformatics. So they're all working together to see its application in health. So we will bring this environmental health dimension in that also. As we are proposing a major center of excellence in AI for health. So we will bring all this in that center.
The third, very important, is in the area of cancer. We are working with one of the leading hospitals in Delhi. We have made a new platform for diagnosis of lung cancer, where we are mimicking the intelligence of radiologists, and we are trying to correlate the radiomics feature, what we call radiology features, with respect to the genomic mutation. And based upon that, the oncologist is able to prescribe the right medicine. So it's a non-invasive technology. We have tested on 2000, now we want to scale up to different hospitals.
Okay.
Yeah.
Now
Well,
let me.
that actually, I got a comment on that, sorry.
Yeah.
This is actually very exciting, and you probably know that Dr. Bertinelli, who's the former director of the NCI, is now the director of the NIH.
Okay.
And I met with her and worked with her, who's the director of the NCI. So we were talking about a lot of these things and talking about the environmental perturbations that can be influencing the development of cancer. And so if you develop cancer, not just for certain molecular reasons, but if there are environmental exposures, the underlying molecular etiology may be different. And so you'll want to treat those patients in different ways. But it's sufficing to say now that as the director of the NIH, she has a great interest in studying gene by environment effects. So I felt that I was just very happy that she was selected as the NIH director. So those of you interested in cancer, it's
No,
I think
he's
they're a good place.
our cancer specialist, VC Das.
Hello.
We call him father of
Unfortunately, I'm out of cards now.
human papilloma virus.
So you
Oh,
can have
that's
this
very important.
NCI, I have
And he
visited. I have-
worked with the Nobel laureate in Germany for 10 years,
Yeah.
who got Nobel Prize for HPV. And he
Uh-huh.
brought that concept over here. And he was founder director of a cancer institute in ICMR, Indian Council of Medical Research.
Terrific. Well, very, very good.
And he has a big cancer group where not only cancer cervix, breast, the head and neck, oral, pancreatic, liver, all this type of cancer under one roof. So he is going to now look at environmental impact. How does it
Good for you.
carcinogenesis, carcinogen, food.
Sounds good.
Yeah, it's nice. I was busy and I was just in between in your talk. So it is very interesting to know it and
And incidentally, he's a geneticist like you.
Terrific.
He's
Good.
Indian
I'm a
geneticist.
geneticist by birth in the academics from Banaras Hindu University, Varanasi. It's BHU, it is called BHU, the Central University. So they environment and environment and gene interactions. And we know from as a student of genetics and the cancer later on that 80% of the cancer are environmental related. And I was also member of the BRC, Baba Atomic Research Center, DAE, Department of Atomic Energy, that Environmental Mutation Society, that environmental mutation is the major agent.
Just renewed my membership.
Yeah, so has the relation with the cancer. So, and gene and environment interaction is most important to lead to many of the diseases, not only cancer. So that's a very interesting and close to our heart that you talked about, and that to connect with the recent developments of the Human Genome Project and and to the genome, the Cancer Atlas, and to the AI. You said as the science is improving day by day, new fields are emerging. We are connecting the health sciences with that. So this is a great learning, and it's great that you are visiting here. And so our institute is the main program is on cancer. So we have around 17 scientists or faculty. So mainly on the cancer, and then they mainly translation cancer research, which has clinical samples flow from the clinics, which is more easier in India so far so that you get a clinical samples. At the same time, you work on the animals, mice, and then cell lines, of course. But then you have a clinical validations. So we have more than a dozen big hospitals, government and private. So all top hospitals, they are in collaborations in our MOU with our institutes, so that our clinical samples, clinical collaborators give the clinical background that what Dr. Kamal said just now, he could develop because of the clinical samples and clinical collaborations.
Some of the programs you can.
Yeah, so programs is mainly are on cancer and stem cell. The institute is cancer and the
Molecular
molecular
medicine
and stem cell research. So the main three program is on cancer, stem cell, tissue engineering, and stem cell therapeutics. And it is translation cancer research related to the drug development. I will just give a glimpse of that, but also the host pathogen interactions. So understanding how that, for example, tuberculosis is very prevalent in India. So one of the very excellent program runs that how that the tuberculosis develop the resistance and how to overcome that. So this is one of the major program
Well,
running.
multiple drug resistance is a huge problem for humanity.
Yeah.
And so I
So for
encourage
the-
you to
Yes.
do the best you can. Let's tackle that problem. Because it's pretty scary to think that in the relatively near future, antibiotics we're going to be at the point where they're not working. So we got to be innovative on that.
So we are not only in the drug resistance of the bacteria, but major project also runs on the cancer drug resistance, cancer relapse. So there is, as far as I know,
I'm fine, I just whittling away at this.
so far we have no medicine actually, clear-cut medicine
I'm fine.
for drug resistance to overcome in cancer. So relapse, cancer recurrence. So we have recently had I'll just briefly tell that we have a molecule which is already patented, and it is called triple conjugate, where we have the original drug, the chemotherapy drugs. For example, one triple conjugate is with the doxorubicin. Then you have a molecule which is from herbal derived, which has been proved several publications that it has a sensitizing property, that when this cancer cells become resistant, they have the cancer stem cell. They are called cancer stem cells. So they have the property of refluxing out the dead. That the reflux proteins, the transmembrane protein, MBCG2 and many other proteins, which has the property to reflux out the cytotoxic drugs. And that's why
Curcumin.
they, so this is the Indian, the spice derived from Indian spice, turmeric, turmeric.
Yeah,
From there,
turmeric, right.
yeah, so that has been conjugated, chemically conjugated with my
Folate.
clear the
And folate gives the signature of the cancer stem cell.
cancer stem cell and
And
cancer cell.
curcumin tones down the toxic effects of the anti-tumor agent.
And
And this is how.
and also sensitize the cancer stem cell. So when it is three conjugates are,
Please have a chance for Dr. Sim Chauhan.
Well, environment question. Samurthy, this meeting I think is very fruitful.
Rick Woychik, very, very nice to meet you.
It's an honor and pleasure to meet you, and I've been hearing the vibrations from this room next door.
Oh, it's very
And,
exciting science that we're talking about here.
Your visit to Haryana was so good.
It
Everyone
was
was.
terrific.
Yeah,
It was
and
terrific.
This Dr. Gurinder Singh exposomics is something fascinating. We have to learn. much
Well,
more.
we're pretty excited about
Yes,
that too.
Absolutely. That's how you.
Great.
Please, yes, Dr. Gurinder Singh, how are you? You met Dr. Gurinder Singh on Route 5.
I did, absolutely. Good. Second time.
Samurthy, where are you sitting?
At the chair.
Please.
On our way, we kept on talking to him about you, our young chancellor.
You have spent time with our wonderful Professor Kamal Rahman, who is like family.
Always,
and,
always.
it's-
She's a great friend.
Yes.
And and I'm just appreciative for her invitation to visit and actually, if it weren't for her, I wouldn't be here right now. Thank you.
But I'll tell all colleagues to have
Phil,
Dr.
hand over the microphone to you.
To have Dr. Rick with us is such an exciting opportunity. The NIH is one of the biggest hubs of innovation anywhere in the world. Many of you may have visited or collaborated. And NIEHS, how environment is impacting people, I think is one of the most important topics that we can think about. I know that many of you are deeply interested in this topic, but when someone like Dr. Woychik visits, I always think, how can we increase our targets, our goals for the university, for our research centers? So Dr. Selvamurthy, I'm so grateful that you are chairing this discussion. And Dr. Selvamurthy said yesterday that at Haryana, it'll be more of a conference. But here we want to have a closed door brainstorming, that what are the outcomes, how can we learn, what can we do together? So I will keep listening. Dr. Das, where you were saying something, please carry on, and would love to listen in and hear what we do.
just briefly too, because there are several people to chair them.
Dr. Asim Chauhan is our additional president of our foundation and also chancellor of few of our universities in India and abroad, Amity Group of Education Institution. He also looks after our incubator, innovation incubator, huge one.
I noticed that on the way in. Good for
Yes,
you.
you saw
That's,
that.
that's very important.
Yeah.
And also the
Having
venture
incubators,
capital.
Innovative new ideas.
Yes, correct. Yes, we have.
Absolutely.
And he's a product of Boston Business School.
Good for you. That's terrific.
Hereby you.
He's from US.
Yeah. Actually, just as a comment, it's, part of my career I did spend in business. And I realized that, after we cloned agouti and they were, very interested in polycystic kidney disease and obesity. It's, if you really want to have an impact, you have to commercialize some of your findings, your research findings. So patent it and then take it into a commercial space. So as we were driving in, I noticed your incubator. I mentioned to Dr. Robin that you're right on the right track. 750 companies. That's pretty impressive.
About 100 of them. But I did read that you have spent time both in industry at different labs and also in academia. I think this combination is wonderful. And in the scientists here, who here has filed patents? I think everyone here must have filed patents. So I think this is something newer in India, Dr. Woychik, that how do you take from a university intellectual property, protect it, and then commercialize it? But one of the great missions that we would like your advice and help also on how can we do this better. Dr. Selvamurthy, yes, we can continue, I think, Dr.
That's briefly.
Just briefly, I ran it that.
Then I want to hear.
I just told you about the conjugate for the drug for the future drug for the cancer to inhibit the cancer recurrence. So that the molecule for the signature, that folate, it will go to, the drug will be taken to the folate receptor bound to the cancer and cancer stem cells. So once it is into the cell solution, it gets to the cytosol. So through the caveolio clathrin coated pits, it gets into, once it binds with this folate receptor, is expressed highly on this cancer and cancer stem cell. And then when it gets to the, with the cell sap, it will, in the pH, it will break the bond, and the curcumin and the drug will be separated out. And the curcumin will do the sensitization of the cancer cells, means it will suppress that reflux molecules, the protein, the protein which reflux the cytotoxic drugs. So that is suppressed, inhibited, so that cells become sensitive, and then the actual chemotherapy drugs can act on that, and that will kill. That is basically.
So
And that-
what's just a quick question. So what's the basis for targeting this? So what's the basis? I'm not understanding. What's the basis for targeting the stem cell versus other cells? Or are you killing other cells too? It's just that you're making sure that you're killing the cancer stem cell, which is where you want to be.
Because the drug resistance is mainly dependent on the cancer stem cells. We want to target the cancer stem cell as well as cancer cells.
Right.
So they express, they are dividing, so they express high level of folate receptor in their surface.
One
So
more
they-
reason, cancer recurs after surgical removal, chemotherapy, radiotherapy, because stem cell is still there. You are not
fully aware of that.
Yeah, that is the
Yeah, you
reason
gotta
that
get the
he
stem
is
cell.
attacking cancer stem cells,
So
so
the
that.
normal cell will not be, it is not going because they are not dividing.
Got
So
it.
it will not go, it will target it. And it will bind to that, get into that, and then it will sensitize and kill this, that cell itself.
So
Otherwise,
where are you in commercialization of this? Is this in clinical trials?
clinical trial, we are doing the animal trials is going on. We could convince the patent judge with our.
Let me
in
just
vitro
answer.
studies, And it is now
We
patented.
have done the in silico modeling of the drug, because it's very complex to make a triple conjugate of a synthetic molecule, natural molecule, and a folate. So which they have done it first step. Then
And
Seema
that-
Bhatnagar has done
Yeah,
the
he is
synthetic
the
chemistry,
Collaborator chemist.
who had made
synthetic
the
chemist.
conjugate in the form of a drug. We standardized them, then we did in vitro studies on cell lines, cancer cell lines. Now the in vivo studies in animals are on. Once you have the preclinical efficacy and safety studies, it will go to phase 1 clinical.
That
That,
is the stage.
Congratulations.
okay, let's listen to
Great
Monica.
example of collaboration. You
Yeah.
gotta be
This is
working
from
with
biotechnology, biochemist.
biotechnologists
synthetic chemist.
to actually figure out how to attach these different molecules.
Dr. Das is one of the most renowned cancer researchers of India. So we're so
Yeah.
proud of his association and the work he's
I'm
doing
happy
here.
to learn that, sir, the NCI director is now a NIH director. I have visited that place, but now
Monica
cancer
Bernoulli
will be
is
given more
terrific.
importance.
Actually,
She totally understands the importance of studying the environment.
Yeah.
So that's critical, because not all cancer biologists necessarily understand that.
Right, right.
Thank you.
So Rick, what he is now currently doing is to give a projections using AI for health. Where
Which
he,
project you can.
this project which we are making now, that we look at genetic factors, epigenetic factors, lifestyle, environmental factors, there is multifactorial.
Well, that's the exposome, man, elements of the exposome, good for you, you're well ahead of the curve.
Yes, and then we are looking at a projection that next 10 years, what will be the disease burden of cancer because of the multifactorial?
Yeah.
Then how do government handle this in terms of public health? How do we prepare the medical systems, our health delivery system for prevention, control,
Cancer burden.
management, and rehabilitation? So he is looking at.
Yeah, reduce the cancer burden.
That's where we can
Model
join.
that out to be able to-
Even in Lancet, you have seen that they use the regular article comes, the disease burden, to reduce
Now,
the disease burden.
Manoj, can you just mention briefly, he is doing on metastasis and also susceptibility to cancer progression. He works with.
Good evening and most welcome to India. So I have got an opportunity to work as Cedars, David Griffin Medical School, UCLA, with Professor Kepler, who is kind of genomics guy. So we have done a lot of sequencing and kind of genetic mutation. At Amity Institute of Molecular Medicine and Amity Institute of Public Health, we have combinedly look at the phthalates, which are kind of plasticizer, which are present in the environment. We have looked at that how they influence the epigenetics, which is, I think, one of your goal in the expo zones. And then we all
That's very important.
Yes.
and then we also looked at that what kind of mutation these phthalates cause. So they cause mutation in the CYP genes, which are important for the metabolism of the drugs and other these
So
plastics.
these activating mutations?
Yeah, these are activating mutation, and now we are loading these mutation and putting into the normal cell and then trying to take the same phelate and see that whether it get into a carcinogenic mode, and then you can test on drugs and the other thing.
But
So
most
I-
interesting is that just to add that I am involved with this research, this study. This is the EDC, the endocrine disrupting molecules. These are the plasticizers which are present, bisphenol and all, the phthalates, the phthalates glucochemical, they are present in the
The
environment.
alkyl substances,
Yeah.
PFAS,
So,
also. in-
Unfortunately, in India, the PVC and all, which is very commonly used indiscriminately, even including the cosmetics. They are present in the cosmetics also, these talent things, but many people do not know how.
We're well aware of that.
Yeah.
We try our best to.
Yes,
to
it is
work
bad
on
in
that.
US, it is, but here it is very, this
See,
indiscriminate.
Amity did one very important study in this connection. They looked at the hypothalamo-pituitary gonadal axis. How will the heavy metals as well as bisphenol, phthalates, how will it affect this endocrine disruption? Then they looked at in the pregnant women. We measured the blood samples, we actually measured the heavy metals. And also in vitro fertilization clinics, artificial assisted reproductive technology. So we published a paper from Amity that we found that this has a big correlation. So when you are treating, you would also handle the heavy metal toxicity which the pregnant women will have, so that your assisted reproductive technology success rate will be better. So we published a paper.
Another great example of the impact of the environment.
Yes.
So if you're using artificial reproductive technology, you want to maximize your chances that it's gonna work.
That's right, absolutely. Then you
Very
are,
interesting.
This is what where we can collaborate and join hands together and work.
yeah, you can give it to Pooja. She heads the biotechnology, genomics, proteomics, and medical biotechnology. Can you explain, Pooja?
Yeah.
And she herself works on aspergillus fumigators. Yeah, please, Pooja.
Good evening, and it's really nice to listen to you, and very exciting. So my name is Pooja, and I am presently the officiating head of the Biotechnology Institute. It is just a couple of lines about the institute. It's one of the largest biosciences institute in the campus. We have close to 2,000 students and around 90 faculty members who are working in diverse fields, which includes medical research, environmental research, artificial intelligence. We have chemists, we have biologists. So we have an array of people with various, varied experience and
A lot of
expertise.
collaboration all that.
Yes, and it becomes very easy for us to collaborate amongst ourselves, because if we require some expertise, we have it in-house. And we are very open to collaborating with scientists within the country and obviously outside the country as well. So coming to my work, I don't work directly on human diseases, but my work basically is, one of my area is to understand the level of the way microbes function in high pesticide pressure. So what we have done in one of our studies is we have analyzed agricultural fields across India where azole-based pesticides are being used. And we have seen, and we have tried to analyze what is the level of mutation which happens in Aspergillus species, particularly Aspergillus fumigatus. So if we recall the level, drugs which are coming into the market, which are actually antifungal, are very low. We still have only 5 classes of antifungals, which are being, given to the patients. One of the most important one is an azole drug, the pentazole drugs, which are posaconazoles and itraconazoles, etc. And the pesticides which are used, without a rationale are also azole based. So if there is a resistance in azole resistance in a microorganism, then those patients will also not, if you give them an azole drug, they won't be treated. So we are trying to understand how these resistance happen, how the microorganism gets, mutated. What is that region where the mutation is actually happening? Not only in Aspergillus fumigatus, you would be very, I would like to share that we tried after COVID-19, black fungus was, the mucormycosis, Rhizopus delamer, was talked about a lot because a lot of patients died because of the secondary infection of mucormycosis. So we analyzed around 50 samples from hospital, near the hospital areas. We found 14 Rhizopus delamer, of which 8 of them were completely azole resistant. None of the azoles could work on it. So we are now trying to find out what is that region where the resistance is happening, how it is changing the protein sequence, and trying to analyze that. So that's my major work.
Well, that's fascinating. Do you have any sense of what genes are being mutated?
Yes, so we have, in fact, in Indian scenario, the agricultural scenario, we have tested close to 450 soil samples. And we have found that there are unique mutations which are not reported. Most of this work is being done in UK and Denmark. And the resistance which is being, reported by them, we have found unique mutations that are happening in India. That may also be because of the difference in the climatic conditions.
So Pooja, you're good. Let us give a glimpse of all that what we want to do in environmental health. Let us focus that. That is with interest. So let's be
That's
brief.
also connected.
But he will answer.
I think
Also
this is
connected to the
terrific.
environment.
And trying to get at you have mutations in genes, that's gonna give you insights into the resistance pathways.
Yes.
And it'll be interesting to see if in the independent isolates, whether you're actually hitting the same biological networks within the organism. Very interesting work. It's fascinating.
Thank you so much.
And
Deeply important.
this is really related to the environment. Now we move on to Tanujinder. She is the head of our environmental toxicity and management. She looks at climate change, human behavior, health, looks at sleep. She's looking at cognitive health over here.
Thank you and welcome to MAT. In fact, I have been interacting with the NIH director earlier at American Center also. So they were keen on having research on air pollution and impact on cognitive behavior. So we are working on that since Delhi is now very heavily polluted with air pollution is very high. We
Tell me
have
about it. I've been experiencing it.
It's not even about you.
Sadly.
Yeah.
We have a continuous air pollution monitoring station at MIT, university here in campus itself. So we get minute-to-minute data. So one project we propose is impact of air pollution on cognitive behavior and neurodegenerative diseases. This is the one which we have on our mind. Second one is investigation of nano and microplastic presence and assessment on carcinogenic effects through bacterial reverse mutation. So these are the 2 things which we propose to study. But I would like to mention that we are one of the biggest in environment domain in India. MIT has 9 institutes and around 14 programs, right from doctoral to master and bachelor's level. Also, we have projects from every funding agency. You name the funding agency, Ministry of Environment, Forest, Ministry of Earth Sciences. We're also working and studying fox in Antarctic region. So that is also one of our forte to provide our expertise in environmental quality program. We have filed around 12 patents and 6 have been granted. And they are in different arenas.
Thank you, Tanu.
Like
Well good.
I have just one question. This is fascinating. So to what extent are you engaging in the development of these new approach methodologies, NAMs? And I mentioned it because this is an area of focus at the NIH right now, where there's a new program called complementary. So it's complement ARIE. And it's all about developing, starting with iPS cells, induced pluripotent stem cells, and developing 3D microphysiological systems that will mimic, human biology, but using those as toxicology testing. So I don't know, are you involved in any of that type of work?
Yeah, that we are trying to explore, but currently we have worked on basically herbal pesticides. And also, we have also seen the impact of electromagnetic field radiation on DNA damage through tunnel assay chromatography.
What is the full form of complementary? What is the RIE? What is it?
So yeah, now you can ask me, I got a couple of my acronyms, so it's, so the complementary is, it's fundamentally about developing these 3D microphysiological systems to complement animal research. And a little, taking a little bit of jet lag.
Right, no,
I
sorry.
can't remember, but I'll tell you.
But the
I'll
idea
find
is-
out what it
but
is.
it- it-
Organoids kind of
To
thing.
develop 3D.
Well, it's
to
organized, so the
model-
3D microphysiological systems, but what's amenable is you can do high throughput. So you're not confined to an individual animal, you can actually do thousands of these within microtiter dishes. And furthermore, you can use iPS cells from an individual, so you can use populations, different populations of iPS cells. So you can be looking at the influence of genetic background. So you bring in the environmental exposures by putting different chemicals or mixtures of chemicals, and then you're looking at different genetic backgrounds in different individual iPS microphysiological based systems.
Yes, wonderful.
So
Thank you, Dr. Tanu. Dr. Tanu Jindal is also on the board of our institute in San Francisco. We're in the Sustainability Institute in San Francisco, Dr. Murinder Singh.
so where is the institute in San Francisco?
Presidio, the Presidio Park.
Okay.
Presidio Park area.
Sounds good. Well, my time in Pfizer was to run a genetics and genomics research institute in Alameda.
Yes, it was just around the corner.
Across-
Yes,
just across the bay.
Yes.
So,
In Alameda and.
Can we have the mind to shooting and?
Yes.
I'm also a regular visitor of ACS and CTEC meetings in US.
okay, sounds good.
Just you people, just 1 minute, you can just, about your research, both of you. No, just give the mic. Yeah, just can you hand over? Briefly, yeah? And sit down and speak. Thank you.
Good evening, everyone. Good evening, everyone. I hope I'm audible. Yes. So, thank you, sir, for giving me an opportunity to present my research. So, as of course, you are from INIHS, which is related to environmental in health and sciences. So, my take on this is that I work in the field of antimicrobial resistance. And how microbes are related to environment, very pretty much obvious that it's a cause of like global warming is a very common these days, and the microbes are, it's very conducive for microbes to grow under this environment. And my aim is targeting an AMR through theranostics, means therapy as well as diagnostics. So we have developed a rapid bedside assay to be supplied in the hospital for treating immunocompromised patients as well as hospital-acquired sepsis patients to test the biomarkers, the sepsis biomarkers at a very faster rate, so that the suitable antibiotic stewardship can be given to them. Secondly, we have also developed novel bio-nanohybrid conjugates, which can target not just the gram-positive but the gram-negative bacteria as well, and susceptible and susceptible and resistant bacteria as well. So I'm working in these fields. So they're very novel because I am utilizing biomaterials for this. So which are amyloidic proteins, which are otherwise detrimental as they cause neurodegenerative diseases such as Alzheimer's, Parkinson's. But I'm utilizing the mechanical properties of these, the morphological properties of these, and making them as scaffolds for doing a localized nanoparticle onto it, and then doing this antimicrobial activity in a very potent way. So that's what I'm working on.
Interesting. What about C. diff? Do you have any good targets for Clostridium difficile?
I'm not,
remains a very important
Yes.
infection
Yes,
in
yes,
hospitals. And there are really no good drugs targeting C. diff.
yes, yes.
The best solution right now is to do a microbiome transfer.
Yeah.
But it sure would be nice to come up with a good drug to target C. diff.
Sure, sir, sure, sir. Right now, we're targeting, yes, definitely. We're targeting S. aureus and E. coli as of now, but definitely we'll be exploring and researching more on that.
Sounds good. Very interesting.
Thank you so much.
Very innovative approaches to doing this.
Good.
Love it.
Thank you, Dr.
Thank you.
Hello, everyone. Hello, sir. Myself, Dr. Banishri. Actually, I started with immuno-oncology. Basically, why? Because at that time when I started my graduations in clinical trials, immunotherapy is very booming. So we took up the PRRs, actually TLR, toll-like receptors. Then I started working with toll-like receptor, and I found TLR3 because there are groups. Some were saying that TLR3 is not at all very good. It is already causing anti-tumor, so why to work on that? But unfortunately, when I started working, that is good fortune for me, but not for everyone. I found out that no, it is actually causing proliferation of those breast cancer cells. Then we reported that, and we actually found that yes, it is actually causing proliferation. But how? Because if it is resides in the cell, inside the cell in the endoplasmic reticulum, which should cause the apoptosis of the cell, then we came up with a pathway where actually TLR can reside in both the places. It actually can sit in cell surface and can act from inside of the cell. So when it is in the surface of the cell, it is actually binding with the adapter that is MYD88 adapter and causing the proliferation of those cells. The same molecule, when going inside, it is actually binding with another adapter that is TRIP and causing the apoptosis. So we came up with a drug. We coated that with liposome because we know our cell is a bilipid layer membrane. We cannot internalize it so easily. So we came up with a liposomal drug. We coated that with a liposome, and then we found out, yes, actually, when it is going inside the cell, it is killing those. That means it is very much useful in clinics, and we started that work with Chittaranjan Cancer Hospital, collaboration with Chittaranjan Cancer Hospital, and that project is still going on. And we found out that yes, in the patients where this TLR is expressing higher, there we have produced an inhibitor also. We can use that inhibitor, and where it is in the cell surface, we can use these liposomal drugs. We can coat that TLR, it will go inside and cause the apoptosis of those cells. So it will be a bit targeted because we need targeted therapies. Then I started looking upon the macrophage polarization, that how it is affecting on macrophage polarization and TILs. Because everywhere where cancer is expressing higher, this tumor infiltrating lymphocytes are high in every patient. So we looked upon that. Then on this journey, I found out bio-tracker with almost more than 1 hour stability because we know these fluorophores usually photo bleach within seconds, maximum minutes. But I come up with a fluorophore which we can use as a bio-tracker because it is not at all photo bleaching and it is biostable for more than an hour, and we patented that. Means we are in the verge of patenting it. It is with Mumbai, Jalgaon University, Maharashtra. And then when I started my postdoc work, I thought of finding out the genes which are actually important in our Indian population, because we have databases, those are dealing with American, European populations outside India, but we don't know much about our Indian population. And then at that time, there is a paper from ICMR of Dr. Saxena and Sunita Saxena, and she was very much focusing on TNBCs. Then I started looking upon TNBCs. I searched the TCGA and Metabricks database, and from there I found out one oncogene and one tumor suppressor. And then I found out a correlation between that oncogene and a tumor suppressor, that is PML and RB, that in each and every TNBC cases where PML is high, RB is lower. That means this PML, which is actually a tumor suppressor, is acting as a proto-oncogene there. And I correlated that data with my Indian patient data because I I am working at that time, even in my PhD with hospital collaboration. And I found out that yes, in Indian population also, almost 70 to 75% of the data which I have collected, the patient samples, they are showing actually concordance with the TCG and Metabricks data.
Right, you still enthusiasm.
Pretty exciting. Pretty remarkable.
So passionate.
Well, that's good. There's nothing wrong with a little passion in the work that you're doing. That's,
Yeah, to Banushree. Good. So we will have separately. Discussion.
by the way, before I go on, I did look it up. So it's complement animal research in experimentation.
Oh, wonderful.
Complement ARIE. And if you leave it to the people who come up with these acronyms for the programs, but. So take a look at complementary, and this is likely to be a major effort to not only create the 3D microphysiological systems for toxicology testing, but then validating them as useful tools to validate drugs and to validate toxicity testing. So keep an eye on this. It's one of the programs that we launched together with Joni Rudder, who's the director of NCATS, one of the other centers for accelerating translational research. But this is where I think almost all of the institute and center directors were really behind this. It's a critical need in biomedical and environmental health science research. So stay tuned on that. Complementary.
Just with all your permission, few more seconds on oral contraceptives. Actually, yes,
Keep going. This is very exciting.
Actually, here in India, women are unknowingly popping up these oral contraceptives, and these oral contraceptives composed of estrogen, xenoestrogens, estrogens or progestin. These have great impact on environment also, because they are popping it up, releasing in the environment. The water is all containing this progestin and estrogen, and again we are taking that water only. That means we are reducing the reproductive health of that woman, and that woman on mental stress again popping up those drugs without even knowing that why she should take that. So there is a huge mental and physical health disorders or health impact on those women. So as actually you are focusing on environmental part, so that is why I just want to take focus on that oral
It's
contraceptive.
a lot of women, not just those women.
Yes,
So
yes.
it's very, very.
Because there is a Mayo Clinic data also, they are also saying that taking these progestin is very much detrimental. It is actually causing breast cancer, not at all cervical and uterine.
Now, I learned yesterday that India has a non-steroidal contraceptive that women use. You take it once a week. So I'm not sure what the molecular nature of that. Is that an estrogenic?
No, these are basically progestins, progesterones. So the kind of progestin, we call it as levonorgestrel. So I have a bit of work on that, and it actually means it is increasing the tendency of the cells to get much more aggressive. So if you are in a luminal stage, it will make you to proceed towards the triple negative kind of a stage. Means you are not actually triple negative, but those cells will become so much regressive that it will proliferate humongously.
Manushree,
Very exciting and very good work. We'll hear from just a few more people in brief. And then I'm enjoying Dr. Woychik's comments on each one of you, because he's thinking and guiding. So then we would like to hear from Dr. Woychik, what are some of the areas that in India particularly could bring a lot of change? Because even sitting in the US, he has an idea globally, what is the kind of work that could have high impact in India and other parts of the world? And also, what is the vision of NIEHS and NIH, how we can collaborate globally? But Dr. Woychik, you will see that not only here, across all of the universities, we have people who are so passionate, top of the line, and we feel so proud of this army of scientists who are all working. How do we make the country and the world better through research and innovation? So we are proud of all of you. A few more
Dr. Kaushik, briefly, yes,
agriculture and
related to environment.
he's a very senior,
She's the Director General of Amity Foundation for Food and Agriculture.
Thank you so much, sir. Good evening to everyone. I will just stick myself to only the topic which we are discussing. So coming to myself, I work on the pollutants contamination. Pollutants from old generation as well as new generation pollutants. Like as she was mentioning about the estrogen and all that. They are all the part of the new generation pollutants, antibiotics, and all the drugs which are we over consume. They are the part of the new generation pollutants. So I study the impact of the pollutants on the health using the metabolomic approach, like of identifying the biomarkers by which we can identify the exposure to these pollutants, whether through air or through water or through food system. And and analyzing this blood and urea sample. So what are the biomarkers which we can identify to identify the exposure to these contaminants? So this is one. I did my postdoc in the UC Davis with Professor Oliver Feen on metabolomics. They were I learned a lot of good metabolomics approach. So multi-omics approach, we can integrate the genomics and metabolomics all together can be done to this just environmental toxicology as well.
So
And,
using high resolution mass spec?
Yes, the high resolution, yeah.
Very good.
Yes.
That's a very important technology
Yes.
for the exposome.
Export.
Correct.
Very-
For your program, we will
Yes, sir.
in this area.
Yes, sir.
Yeah,
So this
no,
is
it's-
one of, yeah.
What about
So,
epigenetic
and,
marks?
Pardon?
What about epigenetic marks? Have you looking at that?
No, I haven't done past, but we collaborate with the genomic data because so we integrate both the genomic data and the metabolomic data together to identify these type of markers. And then one case study we did with the Norwegian people on studying this POP, the contamination of the Ganges, right from the Gangotri up to the down up to the Kolkata. So we studied all the points for this POP contamination, and this transboundary issue was taken up in this one. How these pesticides are like crossing the boundaries without our knowledge. Like we are not using any pesticide POP in Gangotri region or the Himalayan region, from where they are coming. So we did this study and we found out that it is coming from the from through the air, crossing maybe coming from the as far as from the Japan. So this is where the contamination. So air is not a single body's single nation's commodity. It's a multinational commodity.
Stop at a country's
Yeah.
boundary.
Yeah.
As we discovered actually in the
It's a
United States from the
multinational- so air is a
wildfires in Canada.
Right, correct.
Yes, so air is a multilateral issue. It's not a single country issue. That's one thing. Even the water is also a multilateral issue. So somebody is using the Himalayas in the Tibetan region or the Chinese region, and that water might come to our Indian region.
Thank you, Nutan.
So,
Good, we will interact with you.
Fascinating,
Hridesh,
very, very
Hridesh is working in a very important area, sphingolipids and L-serine can be used for tuberculosis control and management.
Yeah, good to see you actually. So I just want to introduce you with my work that we are setting up in a recently installed Center for Translational Research that the periphery are the TB, cancer and IBD. So the idea where what we are doing, we are trying to re-educate our immune system, which has gone haywire, and to put them back on the job so that, that they can support the host, not the disease causing stuff. And here we are we are using the sphingolipids, S1P, one of the major epigenetic modifier of the tumor cell and the damaged cells. Long non-coding RNA or the cancer sensitivity. On the third aspect, we are taking the cytoplasmic.
Thank goodness, yes. So we've got to get beyond just looking at the messenger RNA
Correct.
producing.
RNA.
The rest of the genome is transcribed,
Correct.
and we have to pay attention to that, so.
We are
Good for
trying,
you.
we are trying to change the cold tumor into the hot one so that we can tackle the tumor much better way, and that's the point. And with this, we are also working on protecting the gut from the radiation stress, the lethal radiation stress. That's one of the environmental calamity which we see, right, mentioned by Silva Murthy sir in the context of the defense also. So we are developing the next generation immune pharmaceutical as the deliverables for the human population. For covering the couple of diseases that I mentioned, which, and we are now slowly going for the neurocognitive area also, which is suppressed, which is largely suppressed in the cancer patient. So
And
that's-
L-serine, he is looking at L-serine.
Yeah, we are, we'll let you know about this story later once it is mature.
Right. But now AKC, you wanted to make your brief remark, and then I will go to public health. And then we can conclude.
Thank you. I was listening to your.
Biswas.
And Mariam. Just for my information, not episomoids.
you are launching a program which you said akin to the human genome program. So you would be collecting a lot of data, and that is at the global level. So just
And actually, it's even more complicated than the Genome Project because the Genome Project was relatively simple. It's C's, T's, A's, and G's. Now we're talking about a highly diverse set of data from sleep to nutrition to COPS and PFAS. And so the challenge is to figure out what are the technological paradigms that we use to address this.
I'm coming to that only because you would be collecting huge data. Collection of data is in itself is a big task, but would you like to share your plans to make sense of that data? Because in omics studies, it is not the collection of the data, but to make the sense of the data for our information one. Number two, you have been working Oak Ridge.
Yep.
And Oak Ridge, was a most important lab in the Manhattan Project. And do you still consider ionizing radiations as one of the major factors in exposomics?
Well, ionizing, we can't ignore it. It's certainly one of the very important environmental variables that can be influencing our biology and our genes. So exposomics is all inclusive. It absolutely contains an element of ionizing radiation, and certainly that was a big part of what we did at the Oak Ridge National Laboratory.
That's why I think
So that's actually when I was there. It's I don't know if any of you know, but the mammalian genetics section, Lee and Bill Russell actually started that. Actually, they got their training at the Jackson Laboratory, and then they were recruited in I think it was 48 to start the mammalian genetics program and to really look at the germ cell effects of ionizing radiation, and they did some pretty remarkable work. So they were my mentors when I was there, and that's when I started developing my connections with environmental health sciences.
So why ask this? In your framework of activities, now you have seen, multidisciplinarity. We are working in various areas. Where you feel that we can fit in your this huge program?
Well, so maybe I can just kind of go in and start thinking about this. There's a lot of really high quality research that's going on that's typical of a high quality university. And congratulations to all of you for doing the types of things that you're doing. Now, if we want to start looking about how do we collaborate globally, and let's take the whole issue of precision environmental health. So that's taking exposomics overlaid with genomics, overlaid with big time data science. So I think the key thing is that when we collect the data, we have to be paying attention to common data elements. So that when you collect your data here, and I collect my data, and people are collecting it in different countries around the world, I mean, right now it's the Wild West. Everyone collects the data in the way that they choose to collect the data because they think that their way of doing it is the best way of doing it.
Correct.
And the net effect is that we have data repositories that are not even close to talking to each other. Okay, we need to fix that. Okay, and in fact, in fact, Michael Chang, who's the director of the National Eye Institute, and I were co-chairs of a committee that we went through, and we're trying to figure out how do we sort out all of the different data repositories that the NIH supports. And then once we get beyond that, we have to be thinking about the global community. We should all be collecting data with common data elements so that your databases seamlessly connect with ours. We don't have to get in bed with each other. We don't have to aggregate everything in the same database. Well, let's create a data infrastructure, a global data infrastructure, and let's start with exposomics, because this is a new field now. So what do we collect? Where do we deposit your data? And what are the technologies that we use to collect the data? So if you look at genetics and genomics, when you collect transcriptomics with microarrays, it's fundamentally different data than if you collect it with RNA-seq. Now I'm biased for RNA-seq because I think the RNA-seq is what it tells you what's in the cell. I mean, the microarrays tells you what you think is in the cell because you put an element on a microarray. So we need to develop those same types of technologies, and then developing the data repositories that have consistency so that they talk across different repository boundaries. And then we need to develop the tools where we get, and that's the AI and the ML part. So what are the tools that give seamless access to your database, to my database, so that the world community can take advantage of this broad assortment of data? And furthermore, it's data that's collected from diverse populations. The populations here in India are just different than the white, Caucasian males and females, primarily males in the United States. We need to fix that. So if we truly want to understand the molecular basis of human disease, it's got to have the genetic diversity. So I think you're just thinking across, how can we be working together? Let's work on some of the fundamentals. Think about how you collect your data. How do I collect my data? And that's what we hope to do with this Exposomics Center. So the center should be reaching out globally and then getting input, listening to all of you, and then coming up with a set of rules on how do we collect the data in a way that it becomes seamlessly integrated. It doesn't mean you have to apply those rules, but if you don't apply the rules, then you're not part of this collective data effort. So then once we have these databases all talking to each other, then you have the you AI and ML guys and gals, you're gonna just lust over developing the tools to get in there and integrate across these data sets. So integrating then across some of the transcriptomic data sets, integrating across the genomics data sets. That's where I think. So just starting out, let's figure out how we can collect our data and integrate it better, and then develop the tools. So one thing that we're doing, there's so much, I should have actually brought my slides and actually give you a presentation, but nevertheless, I'll share my slides with you. But one of the things we're doing is at the NIH, we have the 1 million person cohort. It's called the All of Us cohort. So has anyone heard of the All of Us cohort? So if you haven't, let me encourage you. Google All of Us, NIH. It's a 1 million person cohort. It's designed to specifically reflect all of the genetic diversity inherent in the populations across the United States. And the idea was that you go in and you sequence the genome, you do a complete metabolomics analysis, you do a complete transcriptomics analysis. And what we're doing now is, again, bringing this whole mantra of, if you want to understand disease, you got to factor in environmental exposures. So we're starting off with GIS coordinates. So as many of the environmental health scientists know, if you know where someone lives or where they've lived when they were pregnant, and if you know where someone works, and if you know where they worked in the past, you have a pretty good idea of what their environmental exposures were. And so,
I have a permission, you give one idea, just I'm thinking of that, because I also want to work on economics, what you were telling about, because I work on liver cancer. And in the liver is always, that is exposed by several the genotoxic stresses. Like I expose the different, not only liver cancer cell, but also stromal cell with the viral protein like HBX. Those are all part of exposomics, right? So yeah, so the idea is that you are talking about the exosomics in the broad way. If you think about that is in the micro level, like when one patient is coming into the bed for many cancer or disease or something like that, so the doctors are giving a lot of drugs, and that is also part of exposomics. And there is a correlation with the drugs in the term of the organ failures and the liver and other organ also. That is also very important parameter, how you can think about your way, and in the United States, how this kind of the work is going on. Because I was also in US in Vanderbilt Medical Center, Indom Cancer Center, where Paul Mojer was director. So we talk about that, but it's very nice to know about from you. Because here in the MIT, I'm working on the liver cancer.
Did you work with Josh Denny when he was at Vanderbilt?
Yes.
Okay, so Josh, as you probably know, is the CEO of the All of Us program.
Yes.
So
That's what I have.
NIH is tapping into the excellent work that was done at Vanderbilt, probably in collaboration with you.
Yeah, because I'm attached with the pit and pull one.
So you know about balance between food. So I'm attached with her.
I have one question.
Yeah.
So Center for exposonic. Is it at the education or is it a consortium? How does it work?
So it's, well, so I should also just clarify that it's a grant, it's an RFI that we put out there. So we haven't awarded it yet.
Right.
We have a number of proposals that came in. And this will not be at NIEHS, this will be in the extramural community.
Right.
So this will be funded at one of the universities in the probably in the United States. But again, it's, you'll see it on my slides. So there are three different primary areas of focus. And one of them is to develop one of them is to develop the international collaborations on how to operationalize exposomics. So what data do we collect? What are the technologies we use to collect this? And then how do we operationalize this? So when you collect your data on your populations, you'll have genome sequence there. And you'll have, so you can identify some of the unique biological characteristics of individuals within the populations in India. So you overlay that now with environmental exposures. So that's the key. That's where we want to be. So to answer your question, it's in the extramural community. Stay tuned. We'll probably make the announcement sometime in about the June timeframe.
Right.
And that center is going to be charged with really international cooperation. Now, I'll also tell you that, this concept of exposomics is taking root, and it's very quickly germinating across Europe. And so we want to make sure that the things that are germinating in Europe and in Japan, I'm going to Japan in June. We want to make sure that when these things germinate, they're germinating to create a very similar type of plant. So so we have this opportunity. Let's get people collaborating, developing data around common data elements, creating databases that seamlessly talk with each other, all in the spirit of collaboration and doing it right at the outset. So that's where I think the genomics community really failed. So when we started with the Genome Project, it started with a bold vision, and then everyone immediately got out there and started competing with each other. You probably remember
Craig
that.
Venter versus,
Craig Venter versus Francis Collins. But you had also, just within the United States, you had Eric Lander at the Whitehead Institute competing with the folks at Baylor, competing with the folks at Wash U. I mean, this is nuts. So finally, once the genomics community realized that we're never going to achieve the ultimate goal of developing this draft of the human genome if we keep competing with each other. So once the group started collaborating with each other, and that's to Francis Collins' credit, he was the one that basically came in and said, look, if you're funded by the NIH, you are going to share your data, and you're going to do it immediately and around a set of standards. And so once that happened, then the collaboration ultimately facilitated the end product.
Is there any particular slide you want to show, because this is what you presented today earlier?
Oh, great. So this is good. So actually, why don't we start with this?
Yes.
So this is so let me just finish up on this whole notion of, in the lower right-hand corner, the computational biology and data science, getting that right. How do we share data? And then, it's also agreed upon this notion that there's I'll call it the pre-competitive data. So when things come off the genome sequencer, or when you collect primary exposomics data, that becomes enormously valuable when you share it with others. So when you share it with others, that means that you get access to their data as well as your own data. So this is a cultural shift within the biological community. It's especially amongst epidemiologists. So sorry if the epidemiologists in the group, but there's a sense that, I spent all this time developing my cohorts, I'm not going to share my data until I've maximally exploited this. Well, again, I use the model of the genome. So once we started sharing data seamlessly, getting it out there right away, then the magic started happening. So let's use that same paradigm. So let's develop these standards for how to collect it.
It looks to me more like a SFB or ERANET kind of platform as in Europe, ERANET, URIT program. So the Europe has a URIT program, SFP, Special Force Prime. So there's a network of the group working together for the same platform, same mission, you know.
But here the analogy to Project Genome
Yeah, I
is
agree.
very apt.
Yeah.
So that purity of data, homogeneity of data, unless you have a standard form in which you do the data set collections, and the data won't be pure.
Should have a common language and.
Exactly.
Correct.
Common,
So-
so common data elements, CDEs, we're talking a lot about this across the NIH right now. And it's, just biologists, traditionally we grew up, as single labs, you got a PI, you got a postdoc, you got a biologist, and you share your data when you publish things. And then all of these large omic data sets that you collect, you keep on your own computer system, and when you retire, they get deleted. That's wrong. So we need to figure out the ways, how do we collect it? How do we standardize this? How do we quality control it so that when we share it, you don't want to just share garbage. You want to share high quality data. So we're talking about how do we do these things, but changing the culture where you willfully, share your data. And I've been through this. I mean, I've lived this paradigm where if you share your data, you get access then to lots and lots of other data sets. And that and it's changed the way that I've done my science. And the people who I'm working with, they're getting access to my data, it changed the science that they're doing. So that's what we're trying to do, is really changing that it's a cultural shift, sharing the data, just like what happened with the Genome Project.
Yes.
So we can do this now with exposomes.
We'll be with you. We are with you.
Well, that's great.
So we have done this kind of a project where we have 100+ annotator working on different data set coming from the vaccine project, where we were, having people working during COVID-19, extracting the molecules and building the maps out of the network. So I think we need to have a common working group, and we need to establish the standards, and they need to meet regularly to establish the infrastructure where the data would be stored. Then we need a curator team, and also we have extensive experience.
Well, yes, the other thing I'm going to say is that I've got a lot to say on that computational biology and data science. So what we have to do is also shift the paradigm. So right now we all collect data in whatever way that we want to collect it. And then we employ armies of postdocs and PhD scientists to standardize it so that it all talks to each other. I mean, I think what we need to be doing is to changing the way that we collect the data, so that we collect the data in a way that is more seamlessly shareable. And that might involve using different technologies. So an example would be if you use a microarray, or if you use a custom microarray where you're only collecting data of certain genes. I mean, that may be valuable for you, but it's more complicated to share that because your microarray may be different. So how do you integrate two fundamentally heterogeneous data sets? So that's the beauty of RNA-seq. So when you do an RNA-seq experiment, it collects everything that's within the cell. And this is really I have to attribute this to, I don't know, do many of you know Sidney Brenner? The Nobel Prize winner, is very famous. I had the pleasure of working with him in my time in industry. And so he's really the one that invented RNA-seq. And so Sidney's whole approach is that, using microarrays is fundamentally wrong. He said, he lectured me, he said, Rick, he said, microarrays, he said, you're assuming that you know what's going on in the cell. He said, the RNA-seq approach, my approach is you let the cell tell us what's actually happening within the cell. And he's right. So that's why it's using an RNA-seq approach for gathering transcriptomic data. It allows your data to seamlessly integrate with your data. And in fact, when you do an experiment on the same cells as your two data sets are additive rather than two independent replicates of the same data. It's much more powerful. So the bottom line is thinking about how do we collect it in a way that is additive. Rather than just duplicative. So that's something we need to think about. And furthermore, the way we do this, the governance models that we bring to bear on this is that it's not the way Rick thinks we ought to do this. It's we develop governance groups with representation from across the community, with representation from India, from the United States, from European countries. And then the governance groups actually set up the rules and collectively how we want to go about doing this. So it's very, it's a distributed effort and then, there has to be a chair of these groups who ultimately makes the decision. If there's a disagreement, well, you're gonna listen to what people have to say, but then make a decision that this is the standardized way that we're gonna collect the data.
So, Dr. Have you also thought of intellectual property rights when you are accumulating all the data together in a database? How would you give the due
That's a great point. That's a great point. And so I'm old enough to remember the SNP consortium. I don't know, does anyone remember the SNP consortium?
Yes.
So that was back in fact, that was one of my first jobs when I got to Pfizer, was to actually represent the company in the SNP consortium. So the SNP consortium was all about trying to do away with patenting things that are fundamentally not patentable. So you had all of these companies that were patenting SNPs associated with genes, but there was no connection with any biological consequences. So what they were doing was preventing access to this information to the rest of the biomedical community. So the agreement was, as part of the SNP consortium, is that all the pharmas would get together, the NIH was in there, and you all agreed that you essentially put this information into the public domain, which prevents any one organization from patenting it. Then once you have access to the information, then you can farm the data, you can extract knowledge. That's the patentable stuff. And then fine, you do your analysis and you find something that's there's a very interesting SNP connected with type 2 diabetes. You patent that, but that's where there's biological relevance rather than patenting things. So what's emerged is this concept of pre-competitive collaboration. So let's work collaboratively together in a pre-competitive model. But then once we have the databases, let's compete with each other. Okay, you can file your patents, and you should do that. Because as I mentioned, I'm a strong proponent. I think if you don't patent something, the likelihood it's actually going to make it into commercial adoption and dissemination is lower. So patent it, get it into commercial dissemination and get people interested in actually manufacturing it and getting it to hospitals and distributing it to the patients.
Thank you.
I don't know, is any of this making sense? Or
Are
am
you
I just?
comfortable for another 10 minutes? Is it okay, 10 minutes, another 10 minutes? Are you comfortable?
Sure, we can do another 10 minutes. The only thing we need to do is that at some point, my wife is gonna join us for dinner.
Right.
So, any sense of when, so can I pause?
Can you-
Let's go on for 10 more minutes and then we can, we'll invite Janice to join.
We'll also have her here.
Okay, I totally understand.
Where is she at the moment?
Janice,
I wanted to know.
Well, let's keep
the
talking.
hotel or not.
Thanks for the time.
Yes, so she is in Delhi.
Where is she now?
She's at the hotel.
She has reached there.
She's at the hotel waiting for me to let her know when she should, catch some travel to come. I guess she's coming here, is that right?
Which one?
Tell you what, let's go on for 10 more minutes and then we'll figure out what to do with my wife. You know what it is, happy wife, happy life, right? And she's there too.
No, she has enjoyed the sightseeing today in the morning and.
She has, and
A little shopping.
yes. Well, she's not a shopper. She's more of an academic. She likes archaeology and she likes studying. And in fact, she's not a scientist, but she loves the work that we do.
We want to listen to her in case if she comes, we would like to listen to her.
Well, she would actually, I think, be fascinated listening to this discussion.
Thank you. So maybe briefly, public health, Shajina and Shamas. And then we will listen to also Mariam and Rashi. She has published a paper in Nature Genetics and with cardiovascular hypertrophy of the heart.
Right,
You
so-
found some complex
No,
traits.
We were the first lab to come up with the animal model for the hypoplastic left heart syndrome. And because you mentioned Jackson Laboratories, that's where we got all mice from. And
Jackson is a good place.
and I was trained by Dr. Cecilia Lowe, who was the director of genetics and developmental biology. So she's at Pittsburgh and UPMC.
I know her
That's-
work very well. So she's one of our grantees. I know it well.
Yes. So I've been training with her for around 5 years before I moved to India and joined Amity.
And with respect to the environmental, aspects and the public health, how public health can be, I was looking at the role of antioxidants. So two thoughts that came to my mind, like one of the genes, which is the SOD3 gene, which is considered to be protective for COPD. Now, given our environmental conditions over here, I thought something like that, if we look at polymorphisms or SNPs across, or do a GWAS on an Indian population in Delhi NCR, it would be something interesting to look at. And also fortifying food with antioxidants, because, with the Indian economic diversity that we have, not everyone can afford all the things that we talk on the table about antioxidants, and not a lot of people have awareness about it. So fortifying food with antioxidant would be a protective way to, bring it into the natural food or the local food.
I think that's great, and actually supplementing with antioxidants in foods that actually taste good.
Yeah. Yes. Look good and taste good.
That don't taste like medicine.
Yes.
That's your challenge. And I think
We
you're
already
doing
have
good.
the anticonception-rich food with us. So.
Actually, there are many naturally occurring foods
Yeah, we have.
that actually fall into that
We have
category.
the beverage and as well as the food item, both we have antioxidants.
Then we need to collaborate on the public health survey.
Some of those wonderful spices that you have here in India actually function very nicely in
Right,
capacity.
yes. Thank you.
You want,
Very interesting.
Samurti, who next? Someone else.
You also.
Rajshri Das?
Anything?
Rajshri Das? No, Shanas, just briefly.
Okay, so thank you, sir. I will just take 1 minute. So my with the background of statistics, I am working in the area of public health as I did my PhD in population study. I'm also in the collaboration in the list of collaborators in the Global Burden of Disease project at Institute of Health and Evaluation. So in that I'm working here, and as per this environmental project is concerned, I am trying to get something like the forced migration, that means the forced displacement of the people due to the soil and water contamination, particularly groundwater contamination, and the public health burden, and
Can you go to
that and how their life is affecting that, because it's very common in India, pollution is. So on that I would like to work.
No, very good, excellent, terrific.
Greetings, sir. I'm Dr. Rajeshwari Nas from Amity Institute of Biotechnology. Sir, I work on Helicobacter pylori, that's the pathogen that causes gastroduodenal disease. And I'm a part of principal investigator from India on antibiotic susceptibility pattern of H. pylori. And our work has been published in Lancet Gastroenterology and Hepatology, where we found that resistance of H. pylori to clarithromycin is increasing. And that is the treatment for the H. pylori.
Just 1 minute. There is a surveillance, Southeast Asia surveillance mechanism for H. pylori. She is from India, the representative from Amity. She is anchor from Southeast Asia.
That's good. Congratulations, that's quite an honor.
So I was looking into the microplastics. So antibiotic resistance increasing, but in the presence of microplastics, the Helicobacter pylori, it causes biofilm, and that again hindered the therapy of the H. pylori. So I would like to look into the role of microplastics in H. pylori for the antibiotic resistance.
What an interesting application. So one of the, I'm very, quite frankly, sobered by the ubiquity of micro and nanoplastics. But it's just an enormously complex problem because there is no one nanoplastic species. I mean, we're talking, this is even more complex than PFAS, where you have 12,000 different molecular entities. So you have not only the chemical properties of the micro and nanoplastics, but you also potentially have shape, the aggregate forms. And then when you break down the aggregate forms to actually study the chemical constituency, you may be eliminating the biologically active entity. So the microplastics is a very complex issue. I don't know, does anyone ever read my columns? So every month I have something called the Director's Corner. And I try to actually point out the people who are doing some really outstanding work in environmental health sciences. So recently I interviewed Doug Walker at Emory University. He's running one of their exposomic centers there. And we talked, it was a conversational interview. about how do we address the issue of microplastics. So if you're so inclined, you might want to take a look at the director's corner. It's on the NIEHS website. If you can't find it, just send me an
No,
email.
no, it's very easily, I've seen a few of them
Yeah.
and very good.
Yeah.
And Dr. Saramurthy, before we break in a few minutes, it's a very exciting meeting. I want Professor Kamar Rahman to say a few words.
I've
Because
already said.
no, but in my presence, you have to say, because he's someone I have great love and affection for, and you're the reason that Dr. Rick is here. So I would like.
We have an
to say a
MOU.
few words. We have an MOU, but you're someone I have great affection, so Kamar Rahman, you will say a few words. But before she does, Dr. Saramurthy, this word, exposomics, is, I'm hearing this term for the first time, I must confess, and it is new to me. And all the other areas, we already have initiatives, but I'm wondering if at Amity, across our universities, this area of exposomics is something that we should go deeper in, maybe create a cross expertise team, interdisciplinary team to look at exposomics, because it might be an opportunity to take a lead in India. Because we're doing a lot of different work, but how does it, at the end of it, tie in together in a data, systemic data, data-oriented way on the impact of the human health and human body and public health. So, because it's so new, it might be something that we should look for. Your comment, is this something worth doing, and then cover up?
Well, certainly, this is a very good suggestion because the expression is very important area, emerging area, and where Amity has got strength. So all that what we need to do is to integrate systematically and then identify the expertise of each one of you and then make us a force multiplier into one force which can work with him, the main exposome center, which is planning. Can we have a Amity exposome center here with you? So with your leadership, so we put all the data, what we are doing, once we standardize the format, then we will see which one to be purified. The data, some of the data may not be.
The right
exact,
CD,
The right kind of format in which we need to submit to the database. So all the we can work out, thank you, sir. We will certainly revert back to you on this, and we will create a boards and then work with you.
Well, I think that's actually a great way to, sorry, if I could just make a
Yes,
comment
absolutely.
before we turn it to you. I think that's a great way to end this meeting. So I'm here to really relay to you that there's this new emerging area. Right? Because it's not just Rick's idea, we're trying to reflect the emerging, exciting themes that are percolating throughout the environmental health sciences and biomedical community. So this exposomics is, let's think of this as just like genomics 30 years ago. So you're now in a position where you can help to work with us to help operationalizing this. We've got, the exposomics is the old vision. We've got to bring environmental exposures integrated into the way we do biomedical science, because that's the way we're ultimately going to understand the etiology of human disease. So let's do this on a global scale. Let's do it in a collaborative way. Let's bring in lots and lots of innovation. And let's think about how can we take on the challenges of dealing with the totality of exposures and looking at this over the life. So let's bring our collective brains together, and you can bring a lot of unique capabilities in India, synergizing with unique capabilities throughout Europe, throughout Japan, throughout China. That's what we're trying to do, is bring together this global collaborative network.
We'll give a clap for Dr. Woychik.
I'll clap and applaud all of you, because you're going to be doing all the work.
See, India has the philosophy of Vasudhaiva Kutumbakam. So we are all one global family. We are all united. So in this mission also, we will unite.
Sounds good. Well, this is very exciting, and sharing data, let's do it in a pre-competitive space. And then once we have these data infrastructure, then fine, mine it and then patent away and then compete with
And
each
give
other.
your interpretations and application.
Yeah, absolutely. But we want to do this in a way that absolutely promotes biomedical science. So I think that this is a platform. Let's see what we can do. It's again, you're responsible.
Yes.
I'm so appreciative. I'm very excited about being here. And so thank you for the invite and encouraging me to come and visit.
I'm so happy you are here and you are saying that, and thank you very much, Dr. Singh Jahanji. I always say Dr. Gurinder Singh said that you like Asim Chauhanji a lot, that's why you have been taken into Manesar University, but he is here also. And I'm collaborating and I'm giving lectures since 1979 at NIHS. And I'm happy when I met you also, and Dr. Olden has visited us. Dr. Linda Belbon has visited us, and now you are here. And I'm very happy today on a very important subject. We have agreed and we must work on those subjects. So thank you very much. And we are already having our MOU. In your presence, we have to renew the MOU we have.
Yes.
I signed with Linda Belbon.
Clap for Kamara Paul.
And we organized 2 meetings and 3 we are going to do. One was environmental and occupational lung diseases in Lucknow when Dr. Olden came. And my founder director said, what made you to come to India? He said, because Dr. Rahman is invited. So I'm honored. Second meeting was at Lucknow with Linda Berman, impact of environment on
Women's health, that was very important.
Yes, and it was very successful meeting. And now today is the most successful thing, and we must organize another international meeting. Your presence.
No, we will launch a program.
And we will launch this program because this is a very important subject. And thank you very much.
But I think, Kamara, because you have facilitated, with NIEHS, and the next visit of Dr. Woychik, we would like to confer honorary professorship to Professor Woychik and bring him here. If he's able to accept it. So we will write separately for this.
Thank you so much. So you have to come again. And for that, we will organize an international conference, and in that we will do that.
That's very good. Yes. Okay.
If I'm also around.
Thank you. Yeah, well, I'm very humbled by that. Let's focus, let's get the work done.
Yes.
And we can talk about any awards and those sorts of things. That's not why I do things for awards. I do things.
What do you deserve it?
Well, it's whether I deserve it or not.
Yes.
Let's get this started.
Yes.
Let's work on a global scale.
Yes.
And you, I think, can bring some remarkably capable capabilities to bear on this global effort. Let's just do it right.
Yes.
This time versus learning from the Genome Project.
So with the comments for me to wrap up and just sort of next steps and then.
I think before that, just 1 minute to Dheeraj Sharma, we haven't spoken, so can you just.
Hi, Dr. Sharma, of course, yeah.
He is the Deputy Director General of the Amity Foundation for Science, Technology and Innovation Analysis.
Oh, terrific.
He was in the Ministry of Science and Technology as the Secretary of Technology Development Board with the Government of India.
Very important. Yes,
bravo.
As rightly said, welcome to Amity. Welcome to
Thank
India. And a new topic which you have suggested, exposome and a center of exposomics. We will work on it. We have capabilities and expertise you have seen here. People have described that in detail. I just would like to throw upon one subject or topic of maybe I'm intrigued by this. Whether we can work on this, the climate change and future pandemic predictability. I mean, this is what is a topic which was intriguing in my mind. If we work on this, I mean, this is how we should be looking at things. Thank you very much.
Well, so unfortunately, I didn't have a chance to talk about the NIH-wide climate change and health program. It's multifaceted, and we have, and certainly there are major health effects associated with climate change as it relates to the preparing for the next pandemic, which will happen. It's just inevitable. So that's all part of the whole climate change and health program. So it's nih.gov/climate and health. And if there are elements of what you're interested in that are not clearly written in that document that defines the initiative, let me know. We'll change it. We'll modify that and include some of your ideas into that program. But that is also, again, it's I'm all about collaboration, and it's not about ownership. This isn't an NIH or NIHS ownership. It's about catalyzing global collaborative efforts that advance human health. That's what I'm all about. So Again, I'm humbled with the thought of an award. Let's not focus on the award. Let's focus on getting the science done. And thank you.
So thank you very much. Let me just to half a minute. Thank you very much for all of you. You did a great job and we are very proud of you. Each one of you came out with wonderful ideas to collaborate with him. Compliments to each and every one of you. Now take it forward into a concrete concept proposal, send it to us. So that then we will integrate, we will sit together and then give a shape and submit it to him in terms of what he thinks Amity's participation could be in the Exposome Center, which is a global consortium where our Amity should be one of the major partners in this. This is what founder president Dr. Ashok Chauhan always thinks. Whatever we do, we have to be the best. So we'll be a best contributor to your mission there in the NIEHS. And so please send us the concept note. We will sit together and see how we take it forward. Thank you so much. And thank you so much for you, Dr. Nick. That you have excited, all our scientists are so excited.
Yes.
You know,
Well,
they
you
are
got me
charged.
fired up.
They are fired up, absolutely.
Building up an appetite here.
You are a great motivator, you are a great motivator and inspirer. And also thank you, Dr. Singh, for joining us on this occasion. May I now request you to give a small memento to, as an expression of our love and affection to our visitors.
Thank you.
Very-
Very
Happy Dasa.
Thank you so much, and thank you all for taking time.
Yes. Yes. One. Yes.
Outstanding. Good.
Please hand this over. Small game for.
This is silver brick.
Thank you. Thank you very much. This is,
I
very
laugh you
humble.
like it's.
Yeah, I'm gonna have to buy another bag, take all of these.
We can one photograph, take photograph, सब लोग आ जाए with everyone. So pass it in, take it from there, everyone should
Come
come.
closer, come
Dr.
closer.
Sharma. As much as possible, come.
Who's the cards?
And what we do is, Dr. Woychik, why don't you come here in the middle? I'll just go this side of you. There you go. I think this is.
There we go. How about I stand next to
Yeah,
my
but
great, my friend here,
I don't
Dr.
know how you
Rama.
can come here next to
Next
someone
to
like
me.
that, but there you go, yes. Hey, Gordon.
Yes. Thank you.
It's a historical meeting, exposomics we are starting, you know, so what is right? All of you, thank you so much.
Thanks again.
Real pleasure.
Okay. Very good.
Thank you.
Grab my soda.
Yeah, exposed to new areas, exposed.
TCGA or Via portal, all the genomic data
Yeah.
set and.
Well, that's the idea here, but it's much bigger than TCGA.
But my thinking is that if we contribute in your mission, we should fix one variable so that for that variable, Indian data will be aligning with your variable data in US or other part of the country. So if
I agree.
we fix that, then our data will be unique in terms of Indian data.
The other point I want to emphasize, and you can help me understand this, is that I don't want it to come away from this as Rick's vision or Rick's program.